So hello, today I'm going to present to you Matreex, our compact and interactive visualization tool for large gene families. Okay, a bit stuck. Okay, sounds good. Yes, so gene families and gene family evolution is traditionally visualized through gene trees, so where each leaf here is a different gene in different species, and these gene trees can teach us about gene family evolution. For example, from looking at this gene tree, we can identify two different duplication events that I show here as star, and we can also see that in the dog here, that's present in one part of the duplicated gene subfamilies, the other copy have been lost. So that's what we can understand from gene trees. But nowadays, we have a lot of genomes that are available. And so if you want to visualize the relationship between all the genes in hundreds or thousands of genomes, then it starts looking like this, where you have many leaves, so you can look at the pattern of evolution at short taxonomic range. But if you try to zoom out, then it starts really to be complicated to follow what's happening. So this is here, 3 parts of the same gene trees, and these 11 other prints that I'm showing you here. The other possible way to look at the gene trees is to collapse the less informative branch into just into one leaf. But if we do that, then we lose completely the information about what's happening in this branch, whether there's been loss, duplication, or many copy each species has. So that's what we wanted, why we wanted to develop a visualizer that was able to run a compact view of the gene families while losing the least information possible, and that's Matreex. So Matreex is a command line software written in Python that taken as input a species tree, gene trees that's reconciled with the species tree, and it's must be inputted either as a JSON or imported from some comparative genomics database like Panther or OMA. The output of Matreex is an interactive HTML page that looks like this. So here I'm picking up the different components of the Matreex visualization. First, we have a gene tree that can or not be collapsed, but that's collapsed by default, and where you can see different evolutionary events appearing on it, like for example here the little stars corresponds to duplication. To see what's happening in this collapsed branch, we introduce another part of the visualization, that's the corresponding species tree that I'm showing in here, and that can also be collapsed. You can also provide colors for each species in the species tree. So here it's colored by some ionic clays. And then to make the links between these three, we introduce a phylogenetic profile. That's a summary of the number of copies of this gene subfamily that you find in each of the species in the species tree. So the different, like the darker colors show many copies. So we see there's some gene family expansions. And when there's a 0, like at the right, the gene is completely absent from the species. And we have that for all of the gene families, and that's how a Matreex visualization looks like. All of this is completely interactive, so you can uncollapse any of the subfamily trees whenever you want, in which case it will expand the view over the same principle. You can also see in the different parts of the tree when there's a loss. It's a red cross here on the left. Then you can also, if you're interested in some of the species and not some, you can collapse and collapse some of the species to branch, in which case you will see more information about the species in the clade. Matreex can also be used to compare different gene families that are non-homologous, that can be interesting, for example, to find coevolution pattern. So here I give the example of non-homologous gene families that are all involved in the intraflagellar transport complexes that are really important complex for the functioning of the eukaryotic cilium. And you can see here they share a similar pattern of presence absence, and that's because some of the species we saw here have a cilium, and the other, in which we find none of these genes, don't have cilium. And you can see there is some intermediary transition, and all this visualization allows you to see how the cilium, all this machinery evolved in different species across the tree of life. It's in total more than 5,000 genes, but you can see all of this in this one screen. So Matreex is available on GitHub. You can also install it directly through the Python library package organizer, and then you can, if you're interested in knowing more, there's a publication available. So now I thank you for your attention. I want to thank particularly Victor, who actually is a PhD student that did most of the work I'm presenting here, and I'm open to questions. Thank you.